Your life. Yes.
Good morning to all dear delegates. Today is the day 6 and we are going for session 1 right now. In our today's session 1, our today's speaker is Dr. Sachin Surwase. He is a postdoctoral fellow, the John Hopkins University, USA. Let me introduce Dr. Sachin Surwase to all the audience. Regarding his educational qualification, Dr. Sachin is PhD in Chemical and Biomolecular Engineering, Korea Advanced Institute of Science and Technology, Daejeon, South Korea from 2017 to 2021. He has pursued his M.Pharm in Pharmaceutics from Pune University, India during year 2011 to 2013 and secured first class in M.Pharm. He has completed B.Pharm from Government College of Pharmacy, India during 2007 to 2011 and secured first class. Regarding his work experience, he was a postdoc fellow in biomedical engineering at Johns Hopkins University, USA during 2021-22. He was acted as a visiting researcher at Chemical and Biomolecular Engineering, CAST, South Korea during 2021-22. He was a postdoc fellow at Busan National University, Busan, South Korea during 2021 and 22. He worked as project fellow at Physical and Materials Chemistry Division, CSIR, at NCL, National Chemical Laboratory, India during 2013 and 17. He was also a project trainee at Polymer Science and Engineering CSIR National Chemical Lab, India during year 2012 and 2013. He is actively contributing to various international societies and journals. He is a member of British Society of Nanomedicine, member of Society for RNA Therapeutics USA. He is reviewer at Controlled Release Society annual meeting. He is reviewer in MDPI Pharmaceutics. Dr. Sachin is reviewer editor for Frontiers in Bioengineering and Biotechnology. He has honored with number of awards and achievements. He is a receiver of KEST International Student Scholarship Korea in 2017 to 2021. He got Best Poster Presentation Award in 2016 at National Science Day celebration. He is a receiver of AICT Postgraduate Scholarship in 2011 to 2013. He was qualified as the GPAT examination in 2011. Dr. Sachin has keen research interest in following areas: mucosal immunity for cancer and infection, cancer immunotherapy, and diabetes, overcoming the physico-chemical biological barriers for smart engineered nano and micro carrier, targeted delivery of difficult to administer drugs, protein, DNA, and RNA. Biomimetics, bio-inspired carriers, lipid-based and extracellular vesicles for the delivery of DNA and RNA-based therapeutics. Regarding his publications, he has total 13 publications in various reputed national and international journals, including American Chemical Society and CRS, Controlled Release Society. He has filed different international and domestic patents. His international patent filed on lymphatic transport inspired oral GLP-1 gene therapy with non-viral particles for the treatment of diabetes. He has H-index of 9 and I-10 index of 8 with 316 citations as per Google Scholar. With this brief introduction, I heartily welcome to Dr. Sachin Surwase and also request him to enlighten the audience with his knowledge on the topic overcoming gastrointestinal barriers to the oral delivery of biologicals with nano and microparticle system. So I request Sachin sir to start the session.
Good morning, everyone. Thank you so much for a very kind introduction.
And thank you so much to the organizer for giving this opportunity to talk. And today we are going to talk about overcoming the gastrointestinal barriers for oral delivery of biologicals using nano and microparticle systems. So, to introduce the topic, I would like to mention this slide. So, this is a very important slide about the drug delivery, which divides it into first generation, second generation, and third generation drug delivery systems. So, if we consider like 30 years each, like first generation drug delivery systems from 1950 to 1980, so it consists of basics of control release, such as dissolution, diffusion was developed during this time, and then successful control of physicochemical properties were achieved during these 30 years. The next comes the second generation drug delivery systems, which developed a smart delivery systems based on polymers, hydrogels, and nanoparticles. So during this time, gene delivery was also introduced. However, like the second generation from 1980 until 2010, there were inability to overcome the biological barriers. So here comes the third generation system, which is like kind of future systems from 2010 until 2040. So these systems are called like modulated delivery systems, like which is helpful for poorly soluble drugs, and then peptides and protein drugs, and it may achieve the delivery for more than 6 months, and controlled release of for the non-invasive delivery devices. So this will again with the help of smart polymers and hydrogels, and also it may have potential to overcome the difficult biological barriers like blood-brain barrier. However, like in this time, like in this future drug delivery system, we need to deal with the physicochemical as well as biological barriers. So with these, when we talk about the oral or gastrointestinal systems, so this is the path every drugs or molecules takes when we administer it orally. So most of the pharmaceuticals, they try to make the formulation in an oral form like tablets or capsules, because as we know the advantages of oral drug delivery systems, they are easy to administer, and we don't need to go to the hospitals for injection, and there is a huge patient compliance in this oral route. So as we can see in this figure, um, as shown here, when we administer vaccines or biomolecules or small molecules, they go to the stomach and then they go to the intestine, and then there is a M cells for in case of vaccines, where they play very important roles to take these particulate antigens. And then there is a gut associated lymphoid tissue which helps to make a mucosal immunization after this oral delivery of the vaccines. And also additionally, these orally delivered vaccines have potential to go to the circulation, and they produce a systemic immunization. So this is the only route which can produce mucosal as well as systemic immunity to the vaccines, and thus this is very important or vaccination route. um, However, this route comes with various challenges. Like we are going to see here in this slide, what are the challenges for the oral delivery. So first challenge, of course, is the stomach, where there is a very acidic pH degrades these biologicals or vaccines administered by oral route. So because of the stomach pH is very acidic, like it changes from 1 to 3, and also like as seen here, there are enzymes like pepsin and lipase which also degrades these biologicals. So even we able to pass this stomach barrier, the next barrier comes is the mucosal. So because of it is meant to protect our body from the external factors like bacteria and viruses, so it makes hard to deliver anything through to the epithelial cells, which get trapped, and then it will be cleared with the mucus. So this acts as a very strong barrier for the prevention, but which also makes difficult to administer the vaccines by oral route. So second barrier in the line is the mucus barrier, like we just mentioned. Then even we overcome the mucus, then third barrier is the tight epithelial junction or epithelial barrier, which is impermeable to the macromolecules or particulates vaccine system. Um, So if we wanted to deliver the vaccine, we need to overcome all of these barriers, which makes very difficult. So coming to the next slide, so here we can see when we deliver the vaccine by mucosal route, it needs to go like as seen in the left side figure, mucosal delivery. So there are M cells in the intestine, which allows the particles, like sampling of the particles inside, but the other epithelial cells are almost impermeable. So these are the only cells which can take the vaccines administered by oral route and which are present in the Peyer's patches as shown in this figure. However, like small molecules can be easily taken up by the intestinal delivery, like as seen in the figure B, where drug is, if it is log P is more than 5, then it will easily take a lymphatic route, or if the log P is less than 5, then it can take the blood route. So for the small molecule, oral delivery is very convenient and easy. However, for the biologicals or particulate system, MCell only is the way to get into the body. So the in case of parenteral delivery, like as seen in the C, it will, when we inject the molecules to the parenteral route, then it will be taken into the blood or either into the lymphatic system by dendritic cells. Um, to overcome these barriers, the one we mentioned just now, so we come up with a system which can protect from the stomach as well as overcome the mucus and then also penetrate to the follicle associated epithelial cells or FAE. So, what is follicle associated cells? The cells which are lining these lymphoid follicles, they call follicle associated epithelium, and it also contains the M cells. However, targeting just M cells will not be helpful because there are very less number of M cells in the body, and relying on just M cells will result into failure of the vaccines into the clinical trial. So we need to come up with the alternative approaches. So what can be the alternative here is, instead of targeting just M cells, what about targeting the whole follicle associated epithelium like shown in this figure, so that it can penetrate through the M cell as well as the other regular enterocytes, and then it can taken up by the dendritic cells and then by the mesenteric lymph node, where it will produce the mucosal and systemic immune response. So this is the system we developed here called nanoparticle inside a microparticle oral system, abbreviated as NEMOS. So in this case, alginate microparticles are created, and like as shown here, and then they will be loaded with a chitosan nanoparticles, which are modified or engineered with a follicle associated targeting and penetrating peptide. So, like in detail in this figure, we can see, so figure A is like chitosan, chemical structure of chitosan. Then we attached a PEG to it, which will help to reduce the charge of chitosan, because as we know, chitosan is a cationic polymer and the mucus is a anionic in nature. So because of this anionic nature of the mucus, the particles will be just trapped and will not be penetrated to the until the epithelium. So to avoid that, we need to reduce the charge of chitosan with the help of PEG, which is providing a near neutral charge to the chitosan. So next molecule is the CPP in this case. So as I mentioned, we need to penetrate through the regular enterocytes in addition to the M cells. So we need a cell penetrating peptide here. So we attached a cell penetrating peptide, and then this CPP is attached to a targeting peptide, which can target the follicle associated epithelium or FAE, so that we will not be relying on just M cells, or we will be relying on the whole follicle associated epithelium, which increases the chances of delivering the vaccines or particles to the dendritic cells located at the below the follicle associated epithelium. So we are expecting here the particle will be stable when they are delivered through the mouth or oral route, because of the alginate. Alginate is a pH responsive polymer, which is not degrading in the stomach, but it selectively degrades in the intestine. So then our nanoparticles will be released selectively in the small intestine, where these Peyer's patches and these immune cells are located. So continuing with making the particles. So this is the system we developed, like chitosan blank nanoparticles without any antigen. So to develop a vaccine, we used a model antigen here called ovalbumin or OVA. Then that model antigen or protein will be loaded inside chitosan nanoparticles. And next, they will be modified with PEG, as I mentioned, for the mucus penetration. PEG is important. The particle system is characterized for the size, like shown in figure B. Like size is below 160 nanometer for all formulations. The formulation code is mentioned in this case, like formulation 1 is chitosan loaded with ovalbumin. Formulation 2 is chitosan, which is pegylated, which is having a PEG moiety attached to the chitosan for better mucus penetration. Formulation 3 is attached with just RGD peptide, which is just a targeting peptide, not a cell penetrating peptide. And the final formulation 4 is having a PEG plus cell penetrating as well as targeting peptide, CPP-RGD in this case. And it is loaded with the ovalbumin. So as I shown here, like size, polydispersity index was studied for these particles, which was in the acceptable range. And then zeta potential, as we can see after attaching PEG, which is significantly reduced from very highly positive 20 to like less than 10 nanometer. So zeta potential less than 10 is considered as a near neutral, which helps to penetrate the nanoparticles through the mucus. The next is the microparticle characterization. So as we need to load these nanoparticles inside a pH responsive alginate microparticles, so we figure C shows the blank microparticles made up of alginate. Like we can see the figure bar is 200 nanometer. It means particles are around 1 micron in size. And then we can see the chitosan nanoparticle loaded microparticles, which we want to develop. We call it as a Nemos, like because they are nanoparticle inside a microparticle oral system. And then figure D confirms the mucus penetration. Since we are claiming that because of the pegylation, these particles penetrate through the mucus. So we studied this with the help of pig mucus. And then we can see in figure D, the red bar is after pegylation of the chitosan, and the blue bar here is the before pegylation. So chitosan before pegylation was not penetrating, but after we attached the PEG, it started penetrating through the mucus, and this is because of neutralization of positive charge of the chitosan. So this is the particle characterization. And then next one, we wanted to confirm that these nanoparticles are really loaded inside microparticles. So we performed this TM analysis along with metal mapping, because as we know that chitosan is having a nitrogen element which is absent in the alginate microparticles. So as we can see, the blank microparticles contains carbon, oxygen, but there is no nitrogen. But in case of nanoparticles loaded inside these microparticles, we can see that there is a nitrogen present, which comes from the chitosan. So which confirms that these nanoparticles are loaded inside this microparticle system. And then another confirmation will be visualizing with the confocal microscopy. So in that case, what we did is we tagged this molecule, these particles. For example, red color is the microparticles of alginate which are attached to the rhodamine B isothiocyanate, or we call it RBITC, as shown in the figure B. And then nanoparticles of chitosan are conjugated to the FITC. So it is expected that when we merge together, we can see they are co-localized. So it means these nanoparticles of chitosans are loaded inside these microparticles. So this is how we confirm that this Nemos system for their characterization. Next one is, of course, like the pH responsiveness by in vitro method. So what we are claiming here, this alginate is intact in the acidic pH or pH 1.2, and then it will selectively degrade at the intestinal pH of pH 6.8, releasing these chitosan nanoparticles. So we try to confirm this with the help of TM analysis also. So as we can see in the simulated gastric fluid, pH 1.2 at 30 minutes, no particles are broken, they are entirely intact. and also at the 60-minute, there is no breakage of the particles. They are intact. However, in the simulated intestinal fluid, pH 6.8, as we can see, they are broken and the nanoparticles will be released from these particles, from the microparticles. The next one is about studying the release of the model antigen ovalbumin. So we studied this release into two different conditions, like simulated gastric fluid and simulated intestinal fluid. So as shown in this figure, for 2 hours, these particles were incubated with simulated gastric fluid and there was no release. However, once we start the simulated intestinal fluid exposure of these microparticles, then they start releasing the ova, which is expected and desired effect in this case. The next one is visualizing the particles with in vitro M cell model. In case of oral vaccine, M cell model is very important to study these particles by in vitro methods. So how it will be created? So M cell model is basically created out of co-culturing two different cells. For example, CACO2 cells, which represents the epithelial cells, and then those cells will be grown on the insert for 21 days, like as shown in figure B. Like we check the transepithelial electrical resistance. So TER is important for the CACO2 cell membrane integrity, which confirms that there is a tight junction and the epithelial cells are growing well. So, but once we infect the Raji B cells at day 21, as we can see, this TR started decreasing. It means there is a formation of M cells because of these Raji B cells or B lymphocytes. So after creating these models, like we try to check these particles. So chitosan nanoparticle alone, like as shown in figure A. So what we did is we labeled this chitosan with FITC, and then M cells were labeled with the yellow color UEA or lectin, and then wheat germ agglutinin was labeled to the membrane of the cells. So like we can see, there is no targeting and not penetrating effect when we just use the chitosan nanoparticles with this M cell model system. But in case of chitosan PEG nanoparticles, like in figure A, second figure, there is also no targeting and penetrating effect of these particles. But as soon as we attach the RGT, there is a targeting, which we can see here, like chitosan PEG and RGT nanoparticles can target because of the RGT is well known targeting ligand in this case. And then when we attach cell penetrating peptide, there is a penetration we can see here. However, when we attach this cell penetrating as well as targeting together, then we can see there is a more green color coming downwards. It means there is a more penetration as well as targeting of the particles. So to quantify this, like figure C, we quantified it at different time points by measuring the fluorescence of these particles, and also plotted it into a permeability ratio. So in case of chitosan and chitosan PEG, there is a non-significant penetration. However, in case of RGD, like there is a significant penetration, and our final targeting and penetrating peptide shows greatest penetration as compared to the other groups. So after confirming the in vitro penetration of these particles, next is about dendritic cell targeting ability. So dendritic cells are key coordinator of immunoresponse in the intestinal side as well as at the skin side, because they take the antigens and present it to the T cells and then B cells to production of the antibody as well as cellular immunity by producing the CD4 and CD8 cells. So in this figure A, we again check the cell uptake by dendritic cells of these particles. Like we can see in control, there are no particles. However, in case of the chitosan attached PEG and CPP-RGD nanoparticles with different concentrations, like 5 microgram and then 50 microgram. So depending on the concentration, there is a more green color was observed. It means there is more particle uptake by this dendritic cell. And so this particle uptake was quantified with the help of flow cytometry, like we can see here. And this data confirms the dendritic cell uptake by confocal microscopy as well as flow cytometry data. Next one is about these bone marrow dendritic cell maturations. So to study whether these particles are able to mature the dendritic cells, we try to isolate them from the mouse bone marrow. That's why they call BMDC cells or bone marrow dendritic cells. So we use here different maturation markers like CD40, CD86, and MHC1 and MHC2. So as we can see, the final formulation here shows maturation, significant maturation of these markers in figure upper figure of these graphs. And then figure D shows the production of different cytokines like IL-6, IL-10, and TNF alpha, which is also shown very promising in case of finally modified particles. So after this in vitro bone marrow dendritic cell maturation activation study, next is biodistribution of these particles, because now we wanted to study whether these particles also behave similarly in mouse models or mouse study after in vitro like confirmation. So as we delivered these and we isolated important organs from the mouse, like we can see here, number one is the liver, lung, heart, kidney, spleen, and then the whole GI tract or gastrointestinal tract, because we are interested more whether these particles are localized in the ileum portion of the small intestine, because that's where the Peyer's patches and immune systems are located. So as we can see in chitosan nanoparticle alone, there is no color produced because of these fluorescently labeled particles. But in case of chitosan RGD, since RGD is known to target, there is a some color production, fluorescent color is observed here. But in case of the finally CPP-RGD, there is a larger or highest color of the particles is observed. It means these particles are capable of targeting the ileum part of the intestine in an efficient manner. And the figure like the graphs here are just quantification for the colors in the above picture, which shows that formulation 4, which is our final formulation, shows highest color production, which confirms the biodistribution where it should be expected. Next one is about, then we dissected the Peyer's patches and we imaged with the confocal microscopy. So in this figure, we can see that the PBS is the control where there are no particles. And we can see we have marked here follicle associated epithelium and Peyer's patch region. So in case of chitosan nanoparticles, as we can see in this figure, like particles are mostly in the follicle associated epithelium region and they are not penetrated. But when we attach RGD, then they will be penetrated more as compared to the chitosan alone. However, when we have a CPP RGD, which is the tandem or like engineered peptide, which is cell penetrating as well as targeting peptide, where we can see a more FITC green color inside the Peyer's patches, so which is the expected and desirable effect in this case. The graph also quantifies the mean fluorescence intensities, which shows that the final formulation shows there is a greater number of particle uptake by the Peyer's patches in vivo in the mice model. Next is the vaccination of these particles to produce the immunoglobulin A and immunoglobulin G. So the study we designed, like we have delivered 4 doses in total, like prime dose, then after a week first boost dose, then 2 more boost dose, like second and third boost dose, and then we sample this mice for the tracheal, fecal, and serum samples for the production of antibodies. So advantage of oral route is that it can produce immunoglobulin A, which is not produced by the injection route. So that's the advantage of having a mucosal vaccination, which is unique in the way that it can produce IgA, which is not produced by injection. So as we know that most of the bacteria and viruses infect to the intestine or mucosal route. However, currently available vaccines are injectables. So that's where we will not get the IgA production with the help of currently available injectable vaccines. In this case, if we succeed to make the oral vaccination, then it can produce IgA, which is helpful than in addition to IgG in the serum. So coming to the our data, like as we can see here, the formulations which is having a chitosan PEG and CPP RGD attached to it, those nanoparticles loaded inside the microparticles, like we labeled here as a formulation 6, is producing a immunoglobulin A as well as immunoglobulin G at various sites like fecal, intestinal wash, and tracheal wash, as well as in the serum. um, it produces IgG, like IgG1, IgG2, in addition to the immunoglobulin A. So these results confirm that the vaccine, the strategy we used here to deliver the vaccine by oral route produces successfully the antibodies, and it has a potential to help to create oral vaccines in the future. Yeah, so thank you so much for listening to the presentation.
Thank you so much, sir, for such a wonderful session. Uh, I am requesting delegates to kindly post the questions in the chat box if you have any. Sir, we will wait for 2 minutes for any questions in
Yes.
chat box. And sir, there is one question from Dr. Dattatray Shinkar. What
Yes.
are the challenges for development of the nano formulations? This type of nano formulations.
Yes. So actually, main challenges for this type of vaccines is consistency in the polymer, because as we are using a chitosan here, which is a natural polymer, and depending on the source, there is no consistency for the chitosan. So which makes it very challenging for the translation of this type of vaccines, even though they are very promising. But yeah, it's a problem. Another challenge is scalability of the particles. So since they are the clinic like a bench scale, it is very hard to translate them into the clinical scale or larger scale. So reproducibility is another challenge of this type of particles.
Uh, Dear delegates, any questions more? Sir, one question from Shilpa Harak, madam. Can
Yes.
you tell the advantages of these formulations over the other formulations?
Yes. So currently, like the formulations which are developed for the oral delivery, most of the times, like I mentioned, there are like various barriers. For example, there is a stomach, there is a mucus, and then there is the epithelial barriers. So most of the current based vaccines or formulations, they consider either one or two, but not all together. For example, there are many formulations which just consider protecting from the stomach, and then maybe mucus penetration also they work on. But it is very hard to combine them all together, because without overcoming all of the barriers, it is very hard to translate these type of vaccines into the clinic. So that's the advantage of this study, that it considers all of the barriers together by engineering the particles.
Uh, One more question from Tarannum Patave. Please, sir, focus on the stability of nanoparticles.
Yes.
I
So
think,
yes. Yes, so stability of the particles, like when we prepare these particles, they are very robust, and we can freeze dry and store them for even years. Like I have an experience when I prepared these nanoparticles of chitosan, and also the alginate microparticles. They are very stable for at least year when we lyophilize them and store in the refrigerator.
I think there is no question right now. Uh, Thank you so much, Sachin sir, for giving us such an informative session on nanoparticles and microparticle system.
Thank you so much. Thank you so much for the opportunity. It was very wonderful experience to talk and interact with you people.
Okay. Please accept this certificate of appreciation on behalf of MSG Copper.
Thank you. Thank you. Thank you so much.
Uh, Thanks a lot.
Yes.
Dear delegates, the next section will start sharply at. So I request Sachin sir to please leave the studio, and second session will be starting on at 10 o'clock. Thank you so much. Thanks a lot. Thanks a lot, sir.
Thank you. Thank you.